Um, all right, everyone. Well, thank you very much for inviting me here today. I love the energy behind kelp these days. Obviously, kelps are in dire condition throughout a lot of areas of the world, and the amount of energy, the amount of research focus, and just the passion for it is incredible to see. So I'm happy to talk to everybody today about a line of research in my lab that I've conducted along with a number of my graduate students. So today's talk is really going to be a combination of my own work and a bunch of my former students and colleagues. And I call it the hidden life of kelps because it's going to be about the ecology of kelp microscopic stages. We're talking about the zoospores, the gametophytes, and the embryonic sporophytes, those things that we don't see and we don't think about often when we're heading out. So you can see my list of collaborators down here, but I will go through them in one by one as I get to them. So I just want to start off in posing this basic question that really started my career when I was in graduate school at Moss Landing Marine Labs, starting on my master's degree in Mike Foster's lab. And that is, how do organisms survive stressful environmental periods? And so you can think about this, maybe three basic strategies. One of them, that could be just slow growing and long lived, like a cardone cactus or a redwood trees, and they can just persist during periods and just wait them out. Some of them reduce their metabolic activity. They either go through hibernation or torpor or arrested development. And they can survive, just simply wait them out in this kind of this low metabolic or dormant period. And some of them can truly go dormant. And some of our things are, we can think about copepods, tardigrades, or some of our diatoms, where they really have alternate stages that can become dormant. So let's just think about an example. And this is kind of something that started off my thinking about this. Think about the Baja California Sur Desert, right? Very arid, very dry most of the year, but then it rains. And following the rain, this is following the 1997-98 El Nino, the desert could look like this. And where did everything come from? Well, they came from seed banks, right, that were dormant in the soil and waiting for those rain events. Think about the California chaparral. What happens to the California chaparral? Period. If you've been around California, long enough, they burn. And following a burning, you might see something like this, where all the above ground vegetation is taken out. But soon after that, you see this whole recruitment of new plants that weren't there prior to it. And some of these things were even absent prior to the burning. And these things, again, came from dormant seed banks. And so if you take a look at a seed, like this, you have the seed, but then you have these dense seed coats that are very resistant to environmental conditions, and they could last for years in the soil, waiting for the cues to bring them out of dormancy. And those cues can come from heat that could break that seed coat, allow water to enter, and then you wash away the chemicals in there, the hormones that retard growth and development, and the growth promoting hormones take over. Or you might have them chemical. Liquid smoke will actually do this. You could dump liquid smoke on some of these things, and it will bring them out of dormancy. So one of the things about plant seed banks that we've known for a long time is they allow plant populations to be both annual and opportunistic. The question we're here today is, what about algae, particularly the kelps? Do the kelps have something that's analogous to these seed banks? So think about the kelp life history, which is a group of people who are now on this call because they're interested in kelps. So it's the right audience to talk to. If you look starting in the upper left, they might recruit from these juvenile, small, maybe 1 centimeter tall kelp sporophytes. They grow to the adults. The adults release haploid zoospores, which settle on the benthos, become male and female gametophytes. The males release sperm, fertilize the females, produce our embryonic sporophytes, and which grow back into the macroscopic stages. So I want to talk today about these microscopic stages. We know quite a bit, and you probably have seen a lot of talks about the macroscopic stages. They're the ones we think about a lot. But what about what's everything below this line? So if kelps have a dormant stage, I put that in quotes, stage, must be one of these stages. That is, this is the hidden life. So we can ask which stage would be the one that overwinters for periods. What causes them to come out of dormancy? And then what role do these stages play in kelp persistence, recovery following disturbances, and in establishing patterns of biogeography? So that's where I want to go with this talk today. All right, so I'm going to talk today where I started my graduate career at Moss Landing some decades ago, looking at a Desmarestia ligulata, Desmarestia herbacea. Name change has gone through. This is not a kelp, but it possesses the identical kelp life history. It was a nice test organism. That also, working with one of my former graduate students, Billy Beckley, who's doing her Ph.D. at UC Santa Barbara now, she showed during her master's in my lab that these things could be really important in excluding kelp recruitment through scour and shading. So it's a nice place to start, right? And looking at these, to go, we worked in a place called Stillwater Cove, which is in Carmel Bay, just on the other side of the Monterey Bay Peninsula. And if you're looking here, this is our Pescadero rocks right here. And what you're looking at is a kelp canopy from an aerial photograph. And in this, there's 3 areas that are 1,250-meter clearings that I worked with a former student there, Ross Clark, and a bunch of other people in our lab to exclude all the kelp out of it. We basically simulated a disturbance to the canopy and remove the kelp. And we kept these things out for a period of about 6 years. And what we saw is that, and this is Ross hiding in a dense patch of Desmarestia, where we excluded the Desmarestia or the kelp. Desmarestia could exceed 100% bottom cover. Take your points, go straight down, and you got to go through many, many individuals to get to the bottom. And if you just take a look at some data, and these are data from 1992 to 1997, and the black bars represent Desmarestia percent cover in areas where we took the kelps out, and the green bars represent where we left the canopies intact. What we see is when you exclude the canopies, that is the shading mechanism, Desmarestia explodes. And if you don't, just right adjacent to it in the controls, Desmarestia didn't increase in bottom cover much. Tells us Desmarestia is opportunistic. That's one of the criteria that we think about in terrestrial plant seed banks. Another one is we can look at the timing of Desmarestia recruitment. And this is where we had the black bars represent where we took the lines, the dark ones where we took out all the kelp canopies. We had ones where we had just the pterygophoran canopy left. We had one where we had just the macroscopic canopy, and one where we had both canopies intact, the controls. And what you're looking at, it really took us to take out both canopies to really increase that bottom shading for Desmarestia to increase in cover. But the other thing to note here is that there are periods of the year where Desmarestia is absent. So it's not there all year round. It drops to 0. And if you look at the timing of all this, and we look at when Desmarestia recruits, we know it recruits in the spring, kind of just like the kelps have their most abundant recruitment period in the spring and early summer. And so we can look at day length, and this is simply day length where we model it for the latitude of Stillwater Cove. It's easy to do. And we put out temperature loggers, and we look at temperature loggers from January of 92 through mid about 1997. And the red dots represent the date of first sporophyte appearance. That is when Desmarestia recruits. We were diving these sites multiple times a week through this period, so we know when Desmarestia came in. We know the exact day. And what you're looking at is it tends to recruit on the increasing day lengths. And following that spring transition, when you have upwelling winds kick up, Ekman transport, and you have cold water approaching the coasts from upwelling. And so this tells us that it's annual each year. Second criteria is both opportunistic and annual. So does Desmarestia recruit from a long-lived seed bank? Again, it's both opportunistic and annual, and with the population disappearing each year. To address this, I went in and said, okay, what if we remove that seed bank? So we go in, we scrape the bottom, and then to remove the seed bank, we could put these tents where we could sterilize them with bleach on these tents, leave them down, and we could leave these sterilized patches. And then we could have patches where we didn't sterilize them, but we just removed the competitors, the turf algae, and controls where we did nothing. And so we do this, and first thing you notice when you look at the following spring, here is one of those sterilized patches marked at the corners with our flagging tape. You can see the threaded rod that we bolted this down to, and this is all Desmorestia in the route and the surrounding side, and nothing in the middle. And in fact, if you look at Desmorestia recruitment, and where we just removed the turf, that is removed, now there's no kelp canopies, no turf algae to interfere with it. Desmorestia sporophytes were up to 150 to 200 per quarter square meter. We're talking 600 to 800 per meter squared. Where we had the controls, where we had the turf intact, much more reduced, but still heavy recruitment. And where we had them sterilized, we had no recruitment in any sterilized patch except one of the replicates had one Desmorestia. So what we see is when you remove the seed bank, if we will, the microscopic stages, you shut down recruitment. I will say, for those of you who are curious, we did the tests where we brought back substrates to the laboratory, verified they were sterilized. We seeded them, saw recruitment on these sterilized substrates to know there weren't any residual effects of the bleaching. So it's not due to the bleach; it's due to the fact we removed the gametophytes or the microscopic stages. So that brings up, which stage is it? I kind of gave away the story just a second ago, but we'll go through it anyway. So if there is a seed bank in quotes, what life history stage makes it up? To do this, I went into the lab, got Desmorestia to release, stained it with chalcofluor white or fungifluor. It's a non-vital biostain that binds to the cell wall and can be examined under epifluorescence microscopy. We then put out slide racks with these on them and collected them periodically through the entire winter when Desmorestia was absent, and it looked at them under fluorescence microscopy and determined, can we identify them, the stained ones, and can we identify the life stage? And this is from a paper I did on the methodology where you see stained and unstained gametophytes. They show up pretty well in these studies. So what we found, and this is the Desmorestia life history, it looks very much like the giant kelp life history. It starts as a mature sporophyte, releases zoospores, which are biflagellate, just like kelp zoospores are. They land, retract their flagella, form a cell wall, go through gametogenesis, become male and female gametophytes, go through syngamy or sexual reproduction, just like kelps, produce an embryonic sporophytes and grows. And which microscopic stage was it? Every time we looked, we found it to be the gametophytes. So we determined that, and I say the we, it's the people, all the people I've worked with on this, that the gametophyte was this microscopic stage that was overwintering. So does this seed bank live for a few months? And this has been something that's been debated in the literature among researchers and my colleagues for some time now. How long do these things live in the field? Well, to address this, what I did is I did some things where I seeded boulders. I took boulders out that had the seed bank on them, and I sun bleached half of these boulders. And then I took them back in and I put them out in a very deep, 100-foot or more, so it was dark, kind of like under a kelp canopy, so the Desmorestia wouldn't recruit. It was a half a kilometer or so away from any nearby populations, so hopefully it wouldn't get too much of a spore rain. And we held these things for a couple of years. So you can see kind of unseeded and seeded gametophytes. Here's what they look like in these baskets. You can see the seeded, and the bleached and the unbleached substrates. These supposedly had the seed bank on them. The white ones that were sun bleached didn't. We can then translocate these things up after 1 year, and then after 2 years, and then measure them for sporophyte recruitment. And the take home is after the first year, this is about 3 months in the field, no recruitment on the sterilized ones, heavy recruitment on the other ones. Not surprising, it's the exact same result we got from the bleaching, the sterilization bleach tents. Even almost 2 years later, the second season, we did get some spore rain, but we saw much heavier recruitment on those controls. And if you look at the effect size, the difference between this and 0 and here, it held. So what we came from this is that these gametophytes were actually on there for more than a year, at least 2 seasons. So they were long-lived. So that's where I wanted to start this. Now I want to take this into kelp culture, to the kelps. So this is what launched my career into working with kelp microscopic stages. I From this, I went to UC Santa Cruz. I worked in kelp forest ecology, looking at the 1997-98 El Nino Southern Oscillation, and did big scale patterns of kelp disturbance and recovery. But I never lost this passion, this love of the kelp microscopic stages, and thinking that these things are really important. And so I worked with a colleague, Dr. Gustavo Hernandez at CC Mar in La Paz, and we worked together for several years, and we asked, we're beginning to ask about what's going on in with the microscopic stages. So the first one I want to do is a giant kelp forest in Baja California Sur. Scammons Lagoon, Guerrero Negro, right up here that separates Baja California Sur, right? And this is Puteahenia. This is Villa Tortugas, for those of you who know the area. We worked out of Puteahenia and at Puta San Roque right here, which is near the southern range limit. Southern range limit really comes down to about the north end of Villa Abrejos. But we worked here near the southern range limit. And what we did is just doing surveys over several years. We looked at giant kelp density starting before the 1997-98 ENSO event. And we looked at Eisenia, which is now Aclonia arborea. And we looked at their density. And if you look at the relationship between the black bars, which is Eisenia, and macrocystis density, what we see is before the ENSO, they're both at their normal densities. And during the El Nino, they dropped to 0. Eisenia didn't totally disappear. It's a little more resistant to warm water and big waves. Come back to some of that in a little bit. But macrocystis completely disappeared for a couple of years from this area. Well, Eisenia recovered, and it appeared to exclude macrocystis. At some point, after a couple of years, macrocystis began to recover. It showed big variation among sites. It was recruiting into patches, and it began to exclude Eisenia, such that about 2 years after all of this, these populations returned to their pre-ENSO density. So what was going on during this period here? And we were wondering if Eisenia was actually excluding giant kelp. That is, there's a seed bank on the bottom, and Eisenia is just excluding it. So if that's the truth, we could go in and, like we did in Stillwater, remove the Eisenia from experimental patches and see if this enhances kelp recruitment. Don't add any spore bags, don't do anything else, just remove the competitors. And so we can first look at this, and some of this came from this observation that looking at almost 1,500 quadrats over the course of several years, we looked at the recruitment of the small giant kelp versus Eisenia density. And as Eisenia density increased, giant kelp recruitment dropped, such that by the time we get to really dense patches of Eisenia, giant kelp recruitment went away. So this was kind of some correlative evidence that this might be happening. So to do this, we created some similar experiments that we did up in Stillwater. Here's the Eisenia bed down at Puta San Roque. We went in and we asked, is there a seed bank under this? To do it, we simply excluded it. Here's a diver, if you can see it for scale. And this is a 100 square meter clearing that we replicated 3 times. And we did these clearings, and then we came back the following recruitment year. And the idea was, did we get giant kelp recruitment into these areas where it didn't occur on the outside? So just remove the competitors, the seed bank takes off. My worry here was that, for those of you, this is a very unusual day for this area. This is spectacular visibility. Normally visibility in this area, there's a lot of sediment in the water, it's just a few feet. And so we're kind of coming up there going, oh my gosh, how are we going to find our sites? This is before the kind of GPSs that we had and stuff. We had some older GPSs, but they weren't as, didn't have the same resolution. And I was a graduate student, didn't have any. And so we were like, okay, we tried to use lineups, but it's a shifting sand kind of shoreline. And I'm coming up going, okay, hopefully we can find our sites. Well, as we're coming up on them, this is the area. There's no kelp anywhere along the coast except for 3 perfectly squared 100 square meter clearings where we did this. And if you look at 6 months later, we see macrocystis recruiting into the clearings, but not into the controls. We had 1 recruit into 1 control area. But about after 12 months, this is about a full year after it, we still see much higher recruitment in the clearings than in the controls. So at this point, June of 2002, this is about when giant kelp are starting to come in to patches that were just opening up naturally. kelp's naturally thin. They don't stay at those high densities. Patches open up due to waves, storms, disturbance, and giant kelp was starting to take advantage of these. But certainly we saw all we had to do was remove the competitors, boom, and giant kelp came in. Didn't have to do anything else. So it did tell us that, yeah, there's probably a seed bank under these dense Eisenia patches. So if that's true, I came to to San Diego State, excuse me, and I started working in a variety of things. And one of them was I started returning to this subject of the kelp microscopic stages with my students. And this is Dr. Laura Carney, Dr. Paul Matson, and Dr. Damien Say. And these were three people who worked in my lab as master's students and went on to graduate work after that. And if you look at Laura's work, this is nitrate concentration in Point Loma kelp forest in San Diego. And you can see typically in the spring, we have our upwelling, we have nitrate peaking, dropping down, in the late summer and the fall, peaking again the following spring. And we can look at the period of giant kelp peak zoospore release, which is here. And we look at when they recruit. And there's a disconnect, a delay here. And they tend to recruit when that nitrate level is peaking. And so what Laura did is she repeated some of these sterilizations. She went in, sterilized the substrates, hand cleared them, exact same kind of experiments we did in Stillwater, and looked at kelp recruitment in these. And we can look at kelps like macroscopic, our giant kelp, our oarweed, Laminaria farloii, and our walking kelp, Pterogophora californica. And the first thing to note is if we look at recruitment in the sterilized plots, which are these open circles, it takes 3 to 4 months longer to recruit into these plots than they do into the other plots. Again, we did the tests to make sure this wasn't a residual toxic effect, and it's not. We did the laboratory seeding and saw these things recruited onto sterilized substrates in the lab just as much as they do in the natural. So this is a thing of removing the seed bank delays recruitment because now spores have to land, go through the development, and before they produce sporophytes. So it tells us that, yeah, for a bunch of species, this repeats itself. The kelps probably do have a seed bank, and that enables them to recruit sooner, which is going to give them a competitive advantage. If you have a storm that removes the canopies, and who gets there first kind of begins to take over. Right? Kelps are great at that. They need to get in first to be competitively dominant. So to understand why these things came out of dormancy, Laura went into the lab, seeded these things in a whole bunch of petri dishes, and looked at the differences between light, which is that canopy removal, that increase in the radiance, and increase in nutrients, which comes in with that upwelling. So those two things we have seen so far seem to be important in regulating when things recruit. So to tease those apart, she seeded these, and then we did experiments where we held them for 95 days under limiting conditions, very low light and very low nitrate. So we held them quiescent, dormant. And then we transferred them to 100 micromoles of nitrate and 60 micromoles of light. So we simulated a canopy disturbance and we simulated upwelling. But we did these things in combination. So we transferred them, plus N means plus nitrate, plus L means increased light. And then we measured egg production in the female gametophytes and sporophyte production. And we measured these weekly. And the first thing to notice here is if we look at the two sides of this, the left and the right-hand side, the right-hand side is where we manipulated light, but we kept nutrients limiting. The left-hand side is where we increased nitrate, but we manipulated light, plus and minus. If you didn't give them nutrients, didn't matter what you did with the light. They didn't produce eggs or sporophytes. But on the left side, it didn't really matter what you did with light. They produced eggs within about 20 days. A little bit sooner where you gave them nutrients, not surprising. And they produced sporophytes within 40 to 50 days. A week or two sooner where you gave them nutrients, not surprising. So it appeared that nutrients were the main cause of this. And this is one where I got this, and I had predicted with my student that it would be light based on my experience with Desmarestia. Desmarestia is a little bit different critter. It really takes advantage of the high light. But I my student, Laura, she proved me wrong. And it's really nice to be proven wrong because it was a really clear result that nutrients seem to be the thing, and that upwelling events seem to be what was driving this. So we looked across all these different species, Pterogophora, Plagophycus, Macroscopic, and Laminaria, and we can delay these things for periods of not delayed, 2 weeks, a month, transfer them to non-delay conditions, and then we can look at time it takes to produce sporophytes. So now we're looking at, does the time of delay, if you delay them longer, does it matter? And the take home from this is, the longer you delay, going across the x-axis on all of these, the date for sporophyte appearance, the higher you go up, the longer it takes. The longer you delay, the quicker they recruit. So a longer delay probably allows them to build up some machinery. They're photosynthesized. They do have plastids. It probably gives them a bit of a competitive advantage. So delaying not only appears to allow them to persist during periods of unfavorable conditions, but it might give them advantage when conditions improve to come in faster. And then if you can come in faster, again, you can gain a competitive advantage by growing up above your competitors. So it does seem that delaying does convey an advantage. And if you delay longer, it actually might be a little bit of a more of an advantage. So if that's true, if I've convinced you at least so far that, yeah, Kelps, Desmorestia and Kelps, they do have microscopic stages that can persist on the bottom for extended periods of time, that they could be regulated to come in and out of dormancy based on mostly on nutrients, but light has some of it. Once you give them enough nutrients, maybe light helps. But these things come in, and then that they do have a seed bank, if you will, and these seed banks convey an advantage, not only persisting during bad periods, but also recruiting quickly when conditions improve. We can begin to ask questions like, well, what role do these microscopic stages play in setting patterns of kelp biogeography? And we can think about regional biogeography, big scale, local biogeography based on these disturbance regimes, and what about depth biogeography? So this was Paul Mattson, who was a master's student in my lab in the center. He went on to UC Santa Barbara to get his PhD with Dr. Gretchen Hoffman. And Paul worked with Pterogophora californica and Aclonia arborea, formerly Izemia. And we worked in I was still doing my work in California and Baja on the ENSO effects. I was still taking my labs, spending several weeks, a couple times a year down working in the Baja California Peninsula. And if we look at the distribution of Pterogophora and Aclonia, they both have their northern distribution somewhere around Vancouver Island, way up north. The northern distribution is pretty similar. But when you come down to the south, Pterogophora ends up around Bahia Rosario, a little south of San Quintin, where Aclonia goes down south of Mag Bay. There's a pretty good couple 100-kilometer disparity in where they're allopatric, where Aclonia extends further. And so we were curious why. And so we did collect their adult tissues, and then we measured nutrient concentrations in their tissues. Kelps are really good at storing nutrients in their tissues for a period of a couple of weeks. And they could utilize these if the temperatures get warm and the nutrients in the water drop. They can kind of persist for short periods of time, but not really long periods. And so we looked at these things in these two areas, and we just looking at the variation, the temperature nutrients was not explaining it. The Pterogophora, when it got warm in warm places, Pterogophora's nutrients didn't drop in the tissues. And there wasn't a big difference between the species. We didn't believe that this was due to adults. We were curious about, well, what about the microscopic stages, right? American lobsters on the East Coast can survive the adults can survive in our waters, and here the larvae can't. So we know that, the larvae are often the sensitive ones that do this. So what about the microscopic stages? Maybe they are what are limiting here. So to look at this, we can look at temperature profiles through the year in San Diego in the dark and Bahia Asuncion all the way down where we worked in the southern range in the gray. Yeah, and when everything turns upwelling in the early spring, the oceans do cool. But through a lot of part of the year, Asuncion's much warmer. And we can kind of pick 2 points here, kind of that bound, the lower limit of both, and where the two separate. That is, San Diego stops around 18 degrees, where P Asunción is much warmer. And so we can kind of begin to ask, if we choose these two temperatures as kind of bookends, let's take a look at the microscopic stages, how they behave here, and see, is there a difference in their ecology, where Eisenia would occur in these temperatures, but Pterogophora does not. So went into the lab, we had two temperatures, 12 and 18 degrees, different nutrient levels, going a couple orders of magnitude of nitrate, and over 10 weeks, and we can measure sporophyte production. And we did these in Petri dishes again. And if you look at sporophyte production, the density on the x-axis and the number of weeks on the y-axis, and first thing we know is for Aclonia, they recruit after about 4 weeks. That kind of matches some of the other things we've seen and what other people have found. It takes several weeks for kelps to get up and going before sporophytes begin to be produced. That's not new. But it is pretty cool, is that no matter what temperature you are, Eisenia appeared to recruit just fine in 18 degrees. In fact, it might have done a little bit better in 18 degrees. Aclonia, sorry, I keep saying Eisenia, historical way of thinking about it, but Aclonia appears to be maybe even a little more warm tolerant. But if you look at Pterogophora, after a couple of weeks, Pterogophora takes off. It never recruited in those 18 degrees. Other people have seen kind of similar results. Once you get to about 18 degrees, Pterogophora, the microscopic stages all die. So this regional patterns of biogeography, particularly at that southern end, wasn't due to cold temperatures, but it's certainly at the north end, but certainly at the southern end, it appears Pterogophora was excluded from these warmer areas when Eisenia was not. And that's what we concluded in this. And so no sporophytes in 18 degrees. So what about, another thing asked, what about in local biogeography, like depth? This is Dr. Stacy Fatek. She did her master's degree in my lab, went on UCLA. I think she worked at Peggy Fong up at UCLA. And Stacy worked with elk kelp Pelagophycus pora down here in San Diego. This is the Catalina populations. This is what they look like in San Diego. And so if you look at San Diego, here's Point Loma. And this is the Point Loma kelp forest, perhaps one of the best studied Point Loma kelp forests in the world, thanks to Paul Dayton, Mia Tegner, and the labs out of Scripps. And these here, we established 3 transects. These are 1 kilometer long through the forest. We started the deep end, which is outside of the giant kelp macroscopic forest, where Pelagophycus occurs. We looked in the center of the kelp forest and in the shallows, kind of getting into the egregia forests. Now, one thing to note is that Pelagophycus does not occur in these shallower areas. Occasionally, you'll see an individual, but its populations are really limited to where they form pretty dense forests, particularly subtidally or subsurface in the outer waters. And so we're asking, well, why doesn't Pelagophycus come into the center? Well, we did clearings thinking, okay, maybe it was a competition thing, which wouldn't explain why Pelagophycus occurs in deep, darker water, but we still wanted to know, as a competition. It was not. You remove the competitors, that does not promote Pelagophycus. Not surprising. Again, Pelagophycus seems to be limited to these deeper, darker areas. So what Stacy did was went out, collected Pelagophycus, transplanted them using the simple rope twine, where you take a piece of polypropyl rope, split it, put the holdfast through it, let it seal, nail them to the bottom. Had 100% survival on these things, and they attach to the substrate pretty quickly. It's a great way of doing kelp transplants. Did 10 individuals transplanted from the deep, middle, and shallow. So we did reciprocal transplants where we took them in, took them back out to the deep to make sure we had the experimental side of this done right. And then we did this experiment 3 times. And we measured growth, survival, and photosynthetic stress, because we were thinking about it might be something to do with light here. And if you look at the data from this, the dark lines are the inshore, the red lines are the macroscopic. The ones here are Pelagophycus. We could not get data here because this was a period where we could not dive the shallow waters because of shallow. There was a storm in October, so we couldn't get data there. But take a look at these. There's no difference in the mean stipe length. They're all growing. Didn't matter what you did with them. You took them from deep to shallow, back to deep in the middle. This adult sporophytes grew just the same in these things. So it wasn't the adults. It wasn't the sporophytes doing this. And in fact, if you look at the percent with sori, yeah, it took almost a year for these things to become reproductive. but back into Plagophycus bed where they have to grow a much longer distance. Even after a year, they were not fertile. 100% fertility in the inshore ones, and we reached 100% fertility after about a year. They became reproductive sooner in these things. And in fact, if you leave them in the very deep, it might take 2 to 3 years to become mature. So not only were they growing just as well, they became reproductive sooner because they hit the surface sooner. And so it told us that the adult sporophytes actually might even prefer to be in these shallower waters. So why aren't they? Well, she looked at photosynthetic stress using pulse amplitude modulated fluorometry. We could look at quantum yield, a measure of stress. No difference between the three things. It's not photosynthetic. We can look at rapid light curves, which is if you look at irradiance increasing from dark to bright in the electron transport rate in photosystem 2. We see, and we can measure things like alpha photosynthetic efficiency or light harvesting efficiency in this case, the maximum rate of electron transport, where our things saturate, the light levels they saturate at. The thing to note here is the deep water ones actually had lower values of them all. These things actually were highlight adapted as they grew to the surface. So once again, the more evidence that the adult sporophytes should be in the shallower water. They grow faster, they become reproductive sooner, they have greater photosynthetic light harvesting capabilities. And so they do better. So what about the microscopic stages? So here we went back to the lab, seeded these things in culture dishes at 12 degrees. We kept them under low light levels, about 2 micromoles photons, kind of the deep water stuff. We had some of these in 60 to 20 micromoles, brighter. And then we sampled them for sporophyte production, density, and their photosynthetic state. First thing to note is if you look at the light harvesting capabilities of these microscopic stages, we see under low light, and this is for Plagophycus, you can look at quantum yield, 0.56. At high light, it drops to becoming stressed. Photosynthetic light harvesting efficiency alpha is higher, it drops under high light. Maximum rate of electron transport, it drops. Saturation goes up. This is saturation efficiency, but it's brighter light. So what this tells us is light harvesting really they seem to do better under low light, these microscopic stages. They have higher photosynthetic efficiency alpha and a quantum yield under low light conditions. And here, when we transported these things, eventually all the microscopic stages died within a few days to a couple of weeks after we took these measurements. So even under highlight, nothing survived. And we can say, well, what about is Plagophytes then truly different from Macrocystis? Well, here's our Plagophycus under highlight, the same things you just saw, that efficiency, the same numbers you just saw. Compare them to Macrocystis under the same highlight treatments. Everything's higher. Macrocystis is fine under these highlight conditions. Plagophycus has photosynthetic stress and they die. So it gave us pretty good evidence that it's the microscopic stages just cannot tolerate these highlight conditions. We can look at the spores. These are the swimming zoospores, and we can use a phyto pulse amplitude modulated fluorometer. And we can irradiate these things with kind of highlight levels of 900 micromoles, kind of near the surface, mid-water, and near the benthos. You'll notice these 30 micromoles near the benthos are higher, are at that level of the highlight level where the gametophytes all died. And we know kelp spores are sensitive to high light, but the thing to note is this Plagophycus under 900 all died. And in fact, everybody except the low light ones survived, but Macrocystis did better. So all the Plagophycus under high light levels died under higher lights. And where they survived, and they were, even though it was very instantaneous, this is within 24 hours, they're already beginning to show reduced quantum yields and photosynthetic stress, and ultimately they died. So Macrocystis appears less sensitive to the highlight conditions. All right, and so looking at these spores, and looking at Macrocystis, and now we're going to look at these spores, and I'm going to try to wrap this up in the next 7 minutes or so. We can look at spore dispersal. One thing we note is that as you have the waves go past, their orbital is at the surface. By the time they get to the bottom, their energy is all back and forth. But we were curious about unidirectional current flow. That is, what about currents that might disperse them? And so we thought maybe spores might be transported upward and take advantage of these currents. So Damien put out these spore collectors at different depths where we measured zoospore settlement. We did these and put slides on these, did these in the south middle of North Point Loma, and we put out a bunch of these arrays. And this is what these arrays look like. And so we did this in Point Loma to look at spore settlement to find out where those kelp spores were going. And the take home from this is, no matter where you look, as you move off the bottom, here's the bottom going to the surface, and here is the spores. Here, what we see is that spores drop off as you go up. It slides a little wonky, but we're still getting spores 10 meters off the bottom into those bright light levels. So Damien showed that, yeah, these spores do transport up into these higher light levels where they might take advantage of these currents. If they're up there too long, of course, they get irradiated and die. And you can look at these the depth at the shallow levels, we're up in those 900s, but it's when you get down to the lower levels, you get down to those 30s. So transporting them up exposes them to light. So it might not be such a good thing. We still need a little more evidence on this. So Damien exposed spores to a bunch of different levels of light from 750 to 1,000 micromoles for different times. And the big take home is if you look at spore settlement, as you get brighter and brighter, spore settlement drops off, especially as you expose them for longer periods. Sporophyte production drops off. 8 to 12 hours at these high light levels, everything dies. So even giant kelp is sensitive to these high light levels. It's not just Plagophycus. They need to get up there and they need to get back down. Maybe they happen at night, we don't know. But certainly those spores are dispersed through the water column. Okay. So this work by Mike Graham, looking at spores in shallow water. Mike seeded spores onto these ropes, did this in Stillwater Cove, put out ropes to the water column here, and looked, and here's what these ropes look like on these lines. So we have them at different depths. He did these for different life stages, sporophytes, juvenile, gametophytes, and natural. And this is what recruits look like on these. And if you look at these data here, natural, nothing recruits above about 3 meters. Sporophytes move a little shallow, but no matter what you do, the giant kelp really, it's even sensitive. In the upper reaches of those water columns and those bright levels, they do not recruit. And Mike had some nice work showing that giant kelp itself is recruited in shallow water, the first few meters, due to high light sensitivity. So it's not, again, not just Plagophycus. Okay, I'm going to rush, kind of go through this because I know I'm getting long in the tooth there. I want to leave some questions, but this is a really cool, this was an undergrad in my lab, Kyle Van Meter, who looked at mysid shrimp, and we asked, well, what about spores being grazed on? And we can look at mysid distribution, and this is in the midwater, and mysids are more concentrated at the surface. And if you look at grazing, we can put these things in mysids in dishes with swimming zoospores, and simply ask, do mysids graze the zoospores? And so we can use fluorescence microscopy of their guts here. And the nice thing is, without zoospores, here's a mysid gut that's been starved. Nothing's going on. With mysids, they're loaded with zoospores. Mysids are grazing zoospores, and more so in the upper reaches. And if you look at settled spores, doesn't matter if it's dark or light, where there's no mysids, zoospore settlement is high. Where there are mysids, zoospore settlement is decreased. So these zoospores are being grazed by mysids. So I'm going to skip over this because I got a little long in the tooth. I apologize for that. I'm going to, well, message on climate change. It's a little bit more than I have to go in right now. So I'm going to just go ahead and wrap this up here. And I want to say, give some special thanks. So I leave time for credit to a bunch of questions. I'm happy to answer questions about climate change. My students, Matthew Brown and Priya Shukla did stuff and showed that these things are also sensitive to climate change. But those things can be entire talks on their own. So I'm going to go ahead and leave it there. I'm going to stop sharing. And so I leave time for questions on this.